Sir, now we are in live.
Bapraju, live stream is ready.
Okay, sir. So now we are in live. Okay, sir. Good morning all. Today's speaker Dr. Prashant S. Karkar, distinguished guest following the webinar and participants. Welcome to the second day session of FDP-2 on drug discovery and development. Today's topic, fascination in anti-tubercular drug discovery, newer targets, and newer approaches. Welcome to the second day session of FDP-2 on drug discovery and development. We have with us today Dr. Kumar V.S. Nimani, Director Sir of Sri Vishnu College of Pharmacy, who is having B.Pharm and M.Pharm degree in pharmacology from Andhra University, Visakhapatnam. PhD degree from Naipur Mohali and subsequently postdoc fellowship from McGill University, Montreal, Canada. Now I request our Director Sir to welcome all our delegates, our followers, today's speaker, with few words.
Hello everyone. Welcome to day 2 of Day-2 International Webinar on Drug Discovery and Development. I am thrilled with the overwhelming response in terms of the registrations for the webinar, which is about 1,000 participants. I, on behalf of SCCP Management, Sri K. Vishnu Raju Garu, Chairman, and Sri Raichandran Rajgopal, Vice Chairman, Dr. K. Prasad Garu, the Principal, and staff of SCCP, welcome you all, and thank you for attending this webinar on this COVID pandemic. I also thank all the principals and senior staff who are constantly supporting this webinar series. And I must thank Anil Kumar, Organizing Secretary, and Dr. Sujit Kumar, Program Coordinator, for nicely coordinating the event. I'm delighted and proud that Dr. Prashant Karkar, Professor, Institute of Chemical Technology, Mumbai, has agreed to be the part of this webinar series and giving his expertise and sharing with all of us. As you may be knowing, ICT is the Institute of Chemical Technology, a prestigious institute. In fact, it has got the 4th rank NRF. That means the 4th top institute in pharmacy education in India. And this rank is for the past 3 years. Consistently, they are in the top 4. Coming to Dr. Prashant, is a good friend of mine. As I see him as a good scientist, and his publications and his patents and his consultations are really impressive in regard. I am following his work, and I really like the quality and the quantum of work that he's doing at ICT. He's associated with ICT for the past 1 year. Prior to that, he's with the Narsingh Institute, which is also at another prestigious institute in India for pharmacy education. Prior to that, he's an industry research scientist and moved to the academics to do his own innovative work. He's doing a really remarkable work, and I thank Dr. Prashant and welcome you to this webinar.
Thank you so much.
And as always, thank you. It's our pleasure indeed. So as always, all the participants are encouraging this webinar series and hope that this series is exciting, interesting, and useful to all of you. I once again welcome all the participants for this webinar series, and wish that you all continue to participate in the same numbers for the upcoming seminars as well. So with this thank you note, and welcome Dr. Prashant Karkar for today's webinar series.
Thank you, sir.
So over to Sujit.
Thank you, sir, for your welcoming words. Now, myself, Dr. Sujit Kumar, Associate Professor, Department of Pharmaceutical Chemistry, Vishnu College of Pharmacy, taking the opportunity to introduce our today's distinguished speaker, Dr. Prashant Karkar. So presently is the Professor, Department of Pharmaceutical Sciences and Technology, Institute of Chemical Technology, Mumbai. And he has completed his postdoc from Eugene Applebaum College of Pharmacy and Health Science, USA. PhD and M.Pharm from ICT Mumbai. B.Pharm from NDMBPS College of Pharmacy in Nasik. And it's time to say a few words about his experience. He has served as professor in SBKM's NMIMS Institute, Mumbai. He has also served as senior scientist, Nycomed Pharma Private Limited, Mumbai. Scientist, Piramal Life Sciences Private Limited, Mumbai. To his credit, he has completed many funded projects, and presently he has 2 ongoing projects with him. Preclinical development of novel stem cells inhibitor lead molecule, Godavari Biorefineries Limited, Mumbai, and Advent Chemical Biopharma Limited, Navy, Mumbai. He has published more than 60 papers in various reputed journals having good impact factors. And I am happy to announce that he is a registered patent agent in Indian Patent Office. And he was also awarded as the Best Researcher of the Year 2017-18. Now I forward it to you, sir. Dr. Prashant, sir. Please start your lecture. Thank you.
Thank you so much, sir, for your kind words, and thanks to Dr. Kumar as well for inviting me for this webinar. I think it's very interesting to have such a webinar organized with such eminent speakers. I'm talking about the others. For sure, because whatever you hear in these webinars from these established or distinguished speakers, it's really helpful for the students, for the younger generation to and it's like going a long way in their professional career or professional achievements, right? So with these, I will start with my presentation. Yeah, I hope this is clearly seen. Okay, perfect. Okay, let's maximize it. Okay. Now, as you all can see, the title of my talk is Fascination.
Keep it in the presentation mode, no?
Now,
full mode, presentation
mode. Yeah, it is in the presentation
perfect.
mode.
It's right.
Can you see it properly? Okay. Perfect. Okay.
Yes.
The title of, okay, thank you. The title of my talk is Fascination in Anti-Tubercular Drug Discovery, Newer Targets, Newer Approaches. Now, if you ask me why anti-tubercular drug discovery, and as we go ahead, you will see that there is so much of need, unmet medical need in the anti-tubercular drug discovery area for several reasons, and drug resistance is just one of the major problems. And apart from that, India is the global hub of tuberculosis patients. We were probably the first country to have identified patients which were totally drug resistant to all the anti-tubercular drugs. And in fact, Hinduja Hospital was the first hospital in India to have identified these patients. So around 8 of them, they were identified few years ago. They were resistant to all the available anti-tubercular drugs or used anti-tubercular drugs. So all these patients were quarantined in Sangli in Maharashtra, and eventually all of them, they died because there was no cure or none of the drugs would work. Right. And other important aspect is while I was doing my PhD at ICT back in 2003, like I was about to finish in 2003, that time there was one of the crystal structures of MTB, Mycobacterium tuberculosis, DHF, dihydrofolate reductase, which is an established target in many areas, including cancer as well as infectious diseases. That crystal structure was solved, and I was so fascinated, being a molecular modeler or computer-aided molecular design scientist. I was so fascinated with that. In fact, we used that crystal structure of MTB DHFR and eventually tried to like design some molecules and try to synthesize. But by then or back then, there was no facility as such accessible to us for getting the anti-tubercular drugs tested. So that story just remained unfolded. But then when I shifted to academics, I started this very interest of mine, anti-tubercular drug discovery. I found somebody in South Africa who could help us in testing the anti-tubercular drugs. Initially, we got some drugs tested or some of the molecules tested from NCL with Dr. Dhiman Sarkar's lab. But eventually we shifted to South Africa, like all the MTB testing. And in fact, I'll be talking about my own work, part of like 1 or 2 slides, whatever we have done in anti-tubercular drug discovery. So what I'm trying to tell you is anti-tubercular drug discovery is so very exciting. Apart from whatever unmet medical need in India is there. So while we are talking, every third person is infected, or every fourth person is infected with latent TB. Like if we gather around, like there are 40 people, at least 10 of them would be infected with TB, although it may not be apparent or it may not be active. So it is dangerous, it is bad, and that's exactly why I'm here to talk about what's there in the anti-tubercular drug discovery. And moreover, if you look at the per 100,000 distribution of patients in developed nations versus underdeveloped or developing nations, there is a huge gap. In the sense, say tuberculosis is a disease of developing or underdeveloped nations. So that's where the major pharma companies wouldn't really invest in anti-tubercular drug discovery. We are still using the drugs which were discovered like 40 to 60 years ago. It's just only like in 2012, bedaquiline was introduced, and then there were 2 more drugs which were introduced. There was one last year. So there is a huge gap, a huge technology gap, huge discovery gap in the anti-tubercular drug discovery, and that's where now everyone has kind of awake, come to senses and decided to work on anti-tubercular drug discovery. There have been many more consortia which are essentially contributing to open source drug discovery, and a lot of things are happening. I'll just talk briefly about those. So moving on to my presentation, the first part, obviously, like I start with acknowledgements. I thank Dr. Kumar and all others at Sri Vishnu College of Pharmacy, organizers of this webinar, and of course the scientific community whose work I am presenting here, like the literature reports or whatever is known in the anti-tubercular drug discovery area. Okay. The first part, tuberculosis and us. So what is so big deal about tuberculosis and how it affects us? That's exactly what we are going to talk about. So like I mentioned in the beginning, tuberculosis is bad. It's really, really bad for several reasons. WHO's WHO's Global Tuberculosis Report 2019, it listed tuberculosis amongst the top 10 leading causes of death worldwide. And in fact, if I were talking about tuberculosis a month ago, I would have said that tuberculosis tops the list of infectious diseases, the patients suffering and dying of tuberculosis. But then obviously COVID-19 has taken that place. So I would say tuberculosis is only second to COVID-19 to as an infectious disease to cause the infection and the mortality resulting from that. It is dangerous for sure. And India accounts for about a quarter of the global TB burden. Now, this is the most alarming fact. Every fourth person of tuberculosis is in India. So just like any other disease like diabetes or cancer, we are also the global hub or global capital of tuberculosis. Of course, there are other African countries which have got more numbers, but then if you see in terms of the volume, it's very huge, right? And in India, TB is responsible for the death of every third AIDS patients. AIDS patient. Now, this is where the bad side of TB is, because whenever AIDS happen, the immunity goes down, the tuberculosis resurfaces, and usually 25% of the patients, or 25 to say 40% of the patients who are suffering from AIDS, they eventually develop TB and they die of TB, right? Because the immunity is compromised, and the tuberculous or the tubercle bacilli, they come to full activity, and eventually the person dies. Now again, this is very, very dangerous. The death of every third AIDS patient. So it's not only patients who are suffering from only TB, but the TB HIV co-infection, that's more dangerous. And it's not just TB, there are other comorbidities. It's not just AIDS, there are other comorbidities as well. Now you can see the this particular statement: Union government changed the name of revised National Tuberculosis Control Program, that is RNTCP, to National Tuberculosis Elimination Program, NTEP, on December 30, 2019. Now we have got a very like Indian government has got a very ambitious goal of eliminating TB from India by 2025, right? Whereas the WHO has a goal of eliminating TB from the world, from the globe, in 2035. So you can imagine there's a 10-year gap. And given the size of India and the number of TB patients, you can imagine how Herculean this task is. But then it's always good to have ambitious tasks and work diligently towards it, so that you can achieve at least part of it. TB is dangerous, and like earlier mentioned, 1/4 of the global, every fourth person suffering from TB in the world is in India. So the number of persons who get infected and the number of persons who die of TB in India is huge. It's almost like 1/4 of the global numbers. Now, TB is bad because of several reasons, and particularly these reasons including development of drug resistance. So you've got different versions of drug resistance in TB patients, multiple drug resistance, MDR. Then you've got extensive drug resistance, XDR, and of course, the worst part of it, that is total drug resistance, TDR. Like I said, few cases of TDR were already identified in India. So those strains are still there, and who knows how many patients are really getting infected. Of course, the government has a strict vigilance on the number of drug-resistant TB, and the National Program on Tuberculosis. My sister is actually part of it. So they actually monitor the patients, because the TB therapy is so long. Eventually, these patients, they have to be monitored, because if they do not comply with the therapy, if they live in between, that's where the resistance, the problem of resistance comes in picture. So drug resistance problems, together with HIV co-infection and other comorbidities, which I just told you, out of which type 2 diabetes is the most prevalent. Again, like I said, we are the diabetes capital of the world. We are the, in a way, a capital of TB of the world. And that's where the both of these things go hand in hand. Those who are suffering from type 2 diabetes have got a very high propensity to develop tuberculosis sometime in their lifetime, right, depending on their immune status, age, sex, and all other factors. Other important factors which essentially can help in propagating TB is excessive alcohol use and smoking. Smoking is logical because the lungs are directly infected with pulmonary, like we are talking about pulmonary TB, and if you are smoking, then that makes things really worse, and that's where TB epidemics has so prevalent. So global TB control is hampered due to several facts, and out of which the first one being it continues to largely rely on a 120-year-old microscopy-based diagnostic technique with low sensitivity. Meaning we still have those microscopy like sputum analysis where we check for presence of acid-fast bacilli, that is tuberculosis. Although in recent times, like in last decade or so, many new techniques, new techniques have surfaced, but then their use on the mass scale is still not happening. Eventually, it will happen, but then we are still dependent on this 120-year-old microscopy technique for detection. 100-year-old BCG vaccine with limited effectiveness. Like BCG vaccine, it works best in infants as well as the children for preventing the occurrence of TB. But beyond 25 years of age in adults, eventually it does not it in a way does not prevent the spread of TB. It's been almost 100 years. We still don't know how exactly BCG vaccine works. So forget about other vaccine candidates which are under development, right? So this is another issue. And then, of course, I already told you about, because it's a disease of the developing or underdeveloped nations, the developed nations wouldn't really be interested in developing TB drugs, and that's where there has been a huge technology gap. There has been a huge discovery gap as far as new drug discovery for tuberculosis is concerned, and we are still using the drugs which were invented or discovered back in 1952. Isoniazid, that was invented back in 1952. Streptomycin was invented in 1965. Rifampicin around the same time. We are still using those drugs. So you can imagine how bad it is. Of course, the causative agent of tuberculosis, that is Mycobacterium tuberculosis, which is amongst a variety of pathogens, related pathogens, which is responsible for tuberculosis, that is pulmonary tuberculosis as well as extrapulmonary tuberculosis. I told you about latent TB infection. So 1/4 of the world population is latently infected. Like while we are talking, every fourth person amongst ourselves is infected with tuberculosis. I mean, the disease is probably subclinical or it's not seen because the tubercle bacilli are there in the lungs of that person or those persons, but then the disease is not active because the immunity is not letting it to be active. But eventually, at least 10% of these people will eventually develop disease some point of time in their lifetime. So you can see there is a huge reservoir of TB patients, which will never deplete the number of patients suffering from active TB, and that's the most dangerous part. The causative agents of TB, although we know Mycobacterium tuberculosis, but then there are several related pathogens which eventually cause pulmonary as well as extrapulmonary tuberculosis. So collectively, these causative agents are called as Mycobacterium tuberculosis complex, that is MTBC. And it's a group of 7 closely related pathogens whose nucleotide sequence identity is greater than 99%. If you look at the genome, Mycobacterium genome, which was unraveled back in 1998, it contains around 4.4 million base pairs. COVID-19 or other SARS-CoV-2, whose genome is only 30,000 base pairs. So you can imagine 4.4 million versus COVID-19, that is SARS-CoV-2, it's only 30,000 base pairs. So you can imagine how complex this particular organism is. And as we move ahead, you will see how bad it is. So originally it was an environmental organism, but it actually converted over a period of time during evolution into an obligate pathogen. it acquired its ability to grow inside the host cell, particularly the alveolar macrophages, dendritic cells, or other cells of the immune system, as well as the lung parenchyma, and its ability to transmit directly from host to host. So this particular ability was not acquired through new DNA, but through genetic mutation, and we will see about that in few minutes from now. So this particular slide talks about the spectrum of TB. Like while we are, because TB spreads, usually the point of entry is pulmonary, like inhalational route. So while we are in the crowd, and because so many people are infected, even though with latent TB, so we get exposed to TB every now and then, tubercle bacilli. So whenever it gets entered into through inhalational route into the lungs, into the pulmonary tissue. So the first line of defense, or rather second line of defense, that is innate immune response, it comes into picture, and infection is usually eliminated. But eventually, some people will develop the latent tuberculosis, where the either innate immune response or the acquired immune response fails to remove these bacilli, and it actually gets into the granulomatous tissue, because that's how body tries to isolate the infectious agent, and eventually it stays there forever, like till it waits very patiently. It waits a few years, a decade or more than that, before the immune system is lowered and the disease becomes active. So those patients or those persons who are with granuloma in their lung tissue are known as or those suffering from latent TB infection. Whereas some patients, uh, the tubercle bacilli comes out of granuloma, granulomatous tissue, and eventually, spreads to other parts of the lung. But then, because the numbers of bacilli spreading are very low, so you don't really see the symptoms of the disease. But then you still have the microbacteria, replicating inside the lung tissue, and that's what we call it as subclinical TB disease. In case of active TB disease, our Bollywood films have shown, very prominently earlier. Somebody suffering from TB, so the guy puts a handkerchief, he's coughing, he puts a handkerchief, and there is a blood on that handkerchief. So that's a very prominent sign, blood in the in the exudate. And eventually, you also have fever, you have weight loss, anorexia, and all other prominent symptoms, where the TB is, or the mycobacteria are actively replicating, and sometimes it comes out of the lung tissue and goes to brain or any other part. So virtually every single tissue of human body can be susceptible by TB infection. Some people, some female, they actually get TB of ovaries, and their ovaries are completely destroyed, so that they, eventually cannot, do anything related to reproduction. So TB is dangerous. It can affect any and every organ or tissue of the body, right? So you probably know all of these things, but then maybe taking a closer look at these will help to understand the gravity of the situation. So this is a microscopic view of what exactly happens when TB or tubercle bacilli are inhaled or get entry into the lung. So usually in the alveoli, lung alveoli, there are resident alveolar macrophages. So the moment these tubercle bacilli come in contact with the alveolar tissue, the macrophages, the alveolar macrophages would engulf them. That is also true for other immune system cells, that is monocytes, dendritic cells. They will engulf these mycobacteria or same TB uh, by so-called receptor mediated phagocytosis or endocytosis, and this will form a phagosome. Now mycobacteria is so clever, over a period of time, over millions of years, it has acquired the ability to inhibit the phagosome fusion with the lysosome, because the moment it happens, the lysosomal enzymes would digest the mycobacteria, but it prevents the fusion of phagosome with the lysosome, and it prevents the formation of phagolysosome. So in a way, it's a survival strategy for mycobacteria, and it stays there like forever. Now, eventually, these phagocytes and other immune systems with engulfed mycobacteria, eventually migrate to the lymph nodes of the lung tissue for T cell priming, so that a full-blown cell-mediated immunity can be instituted, including the B cell humoral immunity for making antibodies. It is also postulated that mycobacteria somehow crosses the lung parenchymal tissue, although we don't know for sure how exactly it does so. Otherwise, the phagosome containing macrophages would eventually cross the lung parenchymal epithelial lining, and there will be accumulation of other cells, including the T cells as well as B cell, that is the immune system, and that there will be formation of granuloma in some part of the lung tissue. And so this is the stage where the mycobacteria is dormant. It's not replicating, at least for a few years or few days, few weeks, depending on the immune status of the patient. It stays there under hypometabolic state. It's not really actively replicating. So at least 1/4 of the world population has this kind of situation. So you can imagine how powerful the mycobacteria is in terms of its own survival. It doesn't get cleared by the immune system just like that. And this failed immune response, or the inability of immune system to clear mycobacteria, is eventually responsible for the severity of the disease, right? So it stays there like forever. And when the right time is there, it will grow, it will divide inside the macrophages, and eventually or in the granulomatous tissue, and eventually it will grow in such a large number that the granuloma will burst. And that's where the mycobacteria will gain entry into the blood circulation. From there, it can go to virtually any tissue, including brain, and it can re-enter from the blood inside the respiratory or the lung tissue again, and it can infect the other parts of lung. Suppose if there is infection in the right lung, it can spread to the left lung and all other parts of the lung, and eventually particularly in the interstitial spaces, and that's where the disease is full blown, and you see all the symptoms of the active or pulmonary TB infection. Now, to be very clear, it's only the pulmonary TB which is infectious or which is communicable. The extrapulmonary TB is not communicable, right? So that's very important point one should understand. So although we know mycobacteria, and to give you the statistic, in last 200 years, almost 1 billion people have died of TB. Now there is an estimate that since the human life existed on Earth, almost 75 billion people have taken birth on Earth and then they died, right? So like 75 billion people have come and gone from this Earth, so out of which 1 billion died of TB. So you can imagine how bad the disease is. Although we know other diseases like plague and all, when there was no remedy available, so people used to die in millions. But then the TB is so very dangerous, and at least in recent times, it has become very, very bad. And of course, one of the problems why it happens is the resistance of mycobacteria to the currently used drugs, right? So depending on which drugs the tubercular bacilli is resistant to, we classify patients into drug susceptible TB or suffering from drug susceptible TB, where all drugs would work. When it comes to resistant TB, you have MDR TB, multi-drug resistant TB. So here we specifically classify MTB, which is resistant to at least isoniazid and rifampicin. Now, these are two of the 4 first-line agents. So if MTB is resistant to isoniazid and rifampicin, we call it as MDR TB. Apart from that, you have got ethambutol and pyrazinamide in the first-line therapy. The next category is extensively drug-resistant TB. The MTB is resistant to isoniazid and rifampicin, but also to any fluoroquinolone and any of the 3 injectable second-line aminoglycosides, including streptomycin, kanamycin, and amikacin. So if MTB is resistant to INH, rifampicin, and fluoroquinolones, and any of the 3 injectable second-line aminoglycoside, we call that patient as TDR TB. Now, I don't have to talk about XDR TB. I don't have to talk about TDR TB because every drug like the mycobacteria is resistant to every drug, and God is the only hope, which of course it doesn't really work. I'm just saying it for the sake of saying it. And this I have already mentioned to you Hinduja Hospital in Mumbai was the first hospital to report TB, TDR TB cases in India. It's very close to Dharavi area. It's in fact in Dharavi area only. And like I said, 8 patients were identified. Eventually, they were quarantined in Sangli. One of the girls whose weight was 48 kgs when she was identified to have suffered from TDR TB. When she died after a few months, her weight was 16 kg. So you can imagine how bad it is when it comes to TDR TB. If someone suffers from TDR TB, there's no drug actually, right? So that's why it is so bad. So the pertinent question is what causes drug resistance to TB drugs, right? And then answer to these problems are manifold. Uh, and of course, the first one being MTB develops drug resistance through genetic mutation. Now, what we're trying to do is we are trying to put selective pressure on the MTB. We're trying to kill MTB. And like any other pathogen or any other organism in this world, whenever it is put on selective pressure, it tries to come up with strategies which can help it survive. And that's exactly what MTB has done. Through genetic mutations, it has acquired the ability to survive inside the alveolar macrophages, inside the lung tissue, and persist the infection. There are two major mechanisms of drug resistance. First one is target modification, just like any other microbe. To give you an example, mutant bacterial RNA polymerase eluding rifampicin action. Now, rifampicin acts by inhibiting DNA-dependent RNA polymerase of mycobacteria, MTB, right? So the bacteria develops mutations, or the bacteria in a way introduces mutations in the gene for the DNA dependent RNA polymerase of MTB, and eventually this mutated enzyme is no longer able to bind to rifampicin, so that rifampicin action or it becomes ineffective, and that's how the mycobacteria develops resistance. The next one is defective enzyme, right? So to give you an other example, mutant bacterial catalase which fails to activate isoniazid. Now you all probably know isoniazid is a prodrug which needs activation inside the bacterial cell, that is, and the target of action is INHA. The activated drug inhibits INHA. And if the enzyme which is responsible for this conversion prodrug to active drug is defective, and that's where again the mutation happens, and the catalase the mutated catalase is no longer able to activate isoniazid. So isoniazid, being a prodrug, being an inactive moiety, eventually does not get converted to its active form and eventually does not act, right? And that's where this whole issue arises. So for detection of drug resistance, they use various methodologies. First one being phenotypic methodology, then culture-based, where they test the ability of bacteria to grow in presence of anti-TB drug, which is a typical drug susceptibility testing, which is used for other infectious agents as well. This is very clearly evident. And then there are other methods, including whole genome sequencing and others, or next-gen sequencing, which are molecular based methods, right? So eventually one tries to understand whether the bacteria are resistant to the therapeutic drug, and that's where you classify those patients as MDR TB, XDR TB. Now, usually, the TB therapy in drug susceptible individuals lasts for 6 to 9 months, but then for XDR TB as well as MDR TB, it goes beyond 2 years. So, for more than this period, like for 2 years, the patients, they have to take daily these different doses of drugs. Some of these are injectables, and if you see that those tablets are so huge, the dose is so huge, and that eventually leads to toxicity issues, like you are taking very high amounts of toxic drugs for a very long time, and that's where the patient suffers from toxicity issues, particularly hepatotoxicity resulting from INH, ethambutol, for that matter, rifampicin, right? So it's very dangerous for that matter, right? So to summarize, there are several issues: multi-drug therapy for prolonged duration to treat TB and toxicity issues of the first and second line drugs. diagnostic modalities, vaccination approaches, drug susceptibility testing, we are still dependent on very old techniques. So all of these need revamp. And it's not that people are not inventing or coming up with new things, but it's just that they need to be applied on a mass scale. Even if it's a remote village, if you have got some very hi-fi technology, something like next-gen sequencing, which works best, but as long as you are not able to use it in a remote village, it's of no use, right? Because you need to reach the interiors to be able to identify those patients and treat them in due course of time. Otherwise they will spread the TB. Although the infectivity of TB is so very low, but infectivity in the sense one person can potentially infect 8 to 10 patients, unlike what you see for, say, SARS-CoV-2, which is very, very high. Of course, the drug-resistant strains of TB, that's a major problem. And the presence of comorbidities, because when there is a comorbidity, say HIV, a TB patient suffering from HIV patient suffer from TB, so you have to take antiretrovirals. If you are a diabetic patient and suffering from TB, then you have to take a large number of anti-diabetic drug, insulin sensitizers or whatever. And along with that, you also have to take anti-TB drugs. And that's where this whole thing, whole issue of drug-drug interaction comes into picture. So you are popping up so many pills, and there are substantial drug-drug interactions, which in a way are responsible for reduced efficacy of either the anti-TB drugs or the drugs for the comorbidity. So that's another important issue, and that's where one has to be wary of these issues, particularly those physicians which are treating the TB patients suffering from other comorbidities. So obviously, all of this pessimistic view tells us that we need newer drugs to deal with drug resistance problem in TB patients. Like I told you, if a person is suffering from resistant TB, then he or she will need the therapy for more than 2 years. So we need new drugs, which eventually can reduce the span of the overall therapy. And obviously, because these new drugs are likely to address the drug resistance problem, so they should be more efficacious. And ideally, because there is always a problem of cross resistance, those drugs, they have to be, essentially they should be acting by a different mechanism than the first and the second line drugs, right? So that's an important consideration. So that brings us to the next pertinent question. What are the likely attributes of these newer drugs which are likely to act against the drug-resistant forms of the tubercle bacilli or tubercle patients, in tuberculosis patients? So the lead criteria, 3 important lead criteria for a new anti-MDR TB drug. So we are specifically talking about drugs which will be effective for treating MDR as well as XDR TB. We already have drugs for susceptible TB. The first line and the second line drugs, they work excellently, but it's just that the problem of MDR TB has complicated the situation. We need new drugs to treat MDR and XDR TB, right? And maybe these drugs will eventually also be useful for TDR TB because they're acting by a different mechanism of action. Very important consideration or very important lead criteria. These drugs must possess mechanism of action or mode of action distinct from existing anti-TB drugs. Otherwise, there will be cross-resistance problems. We all know what cross-resistance issues are. Should exhibit a pharmacological profile which suggests the capacity for good tissue penetration with minimal liabilities in terms of post-toxicity. Now, I told you the mycobacteria resides in the granulomatous tissue for a very long time, and usually these areas are not penetrable by the existing anti-tubercular drugs. So you have to have drugs which will have good tissue penetration, which probably can get inside the granulomatous tissue and kill the mycobacteria which is residing in the granulomatous tissue in the alveolar macrophages or other cells of the immune system which have engulfed the mycobacteria, right? And obviously the drugs should be not toxic because you will be eventually taking for a very long time, and that's where you need to have drugs which will be less toxic to the host and should preferably act synergistically with, but at least it should not antagonize existing front and second line anti-TB drugs. I mean, like I told you, The first and second line drugs, they act very nicely. So if you have a new drug, to be able to to delay the resistance to these new drugs, if you eventually have to give them in combination, say with the existing anti-tubercular drug. So if they act synergistically, that is perfect. So at least they should not antagonize the currently used anti-TB drugs. Otherwise, the whole problem will exacerbate. I mean, the problem will not be addressed, right? So this whole thing necessitates the search for novel drug targets with proven potential, old targets with novel mechanism of action. Now, in fact, there are some very interesting examples, including DNA-dependent RNA polymerase for old targets with novel mechanism of action. There are drugs or there are compounds under clinical development which act on the same target but at a different site, right? The DNA-dependent RNA polymerase, it's a very big enzyme. So eventually, these drugs, these new age drugs, inhibit the same target but act at a different site, and there is no cross reactivity with these drugs. There is no cross reactivity with rifampicin, which acts on the same target, and that's where you need to have you can use old targets, but as long as you have drugs acting by a novel mechanism of action, you are good to go. New chemical matter acting against novel drug targets. This is very important. Again, sometimes the resistance could be very specific to the chemical matter or the structure of the molecule itself. Newer in vitro and in vivo models, which will help in screening the libraries of newer compounds, could be phytochemical libraries or could be other natural product libraries. So you have to have high throughput assays, even though you come up with a novel drug target. But if you do not have a high throughput assay, or if the assay is too complicated, that will hamper the development of therapeutics against these particular for the tuberculosis drug discovery. Also, the in vivo models are very important, because in vitro you may get excellent activity by these molecules, new candidates or new chemical entities. But if they do not act in vivo, it's of no use. Like the tissue penetration thing, which I mentioned about. There are an array of host-directed therapies. So it's like if you cannot really kill the mycobacteria, you try and devise strategies which are directed to the host, particularly strengthening the immune system or taking care of some of the complications which the mycobacteria causes, and eventually make our body so strong to be able to kill the mycobacteria. So these are host-directed therapies. Methods for addressing drug resistance issues with clinically used anti-tubercular drugs. So if we know why is drug resistance happening, we are in a better position to address those issues and tackle those issues. So we need all of these things to be able to defend ourselves against the highly dangerous tubercle bacilli. So that brings to the next point of discussion, that is strategies for anti-TB therapeutics. So roughly these strategies are two major strategies. One is host-directed therapies. The other one is pathogen-directed therapies. But again, in pathogen-directed therapies, you have to, you can develop drugs against essential targets essential for the survival of mycobacteria, and then non-lethal targets. Because what happens is, if you're trying to put a selective pressure by putting or by inhibiting the essential targets, the mycobacteria will devise a mechanism sooner or later, maybe through target modification or genetic mutations and other mechanisms. But if you have got non-lethal targets which eventually are involved in the virulence, so if you're trying to, if you reduce the virulence of the mycobacteria, immune system will take care of its clearance, right? So, and in the process, you're putting, because you're inhibiting a non-lethal target, you are putting less selective pressure on mycobacteria, and eventually the process of resistance development will be delayed, compared to the ones which are acting against the essential targets. And of course, the host-directed therapies. So you modulate the response of the host to the mycobacteria and try and take care of the mycobacterial infection. So this is again a closer look at these two strategies, the host-directed strategies and the pathogen-directed strategies. So on the left hand side, you see host directed strategies where you're trying to address various issues which happen in a host due to the underlying mycobacterial infection. So in a way, you're trying to produce or improving or enhancing the autophagy via mTOR inhibition of toll-like receptors. Then you also have you're trying to inhibit the process of granuloma formation. So in a way, what you are trying to do is you're trying to take the mycobacteria out of the granuloma, because the moment it comes out of the granulomatous tissue, the drugs which are acting like the clinically used or currently used drugs, they will clear off the infection. So you can have TNF inhibitors, VEGF inhibitors for interfering with the process of granuloma formation. You also have pro and anti-inflammatory host responses, particularly I'm sure you are aware of cytokine storm, which happens in COVID-19 patients, and people are trying to deal with the cytokine storm, which is majorly responsible for killing the patient because of the excessive immune response. So people are trying to come up with strategies in COVID-19 by taming down the immune system. So similar things can be done here, because the underlying inflammation causes damage to the lung tissue. It becomes fibrous tissue and eventually useless tissue, and that's where the patient eventually loses the lung tissue and eventually dies of the complications. So if you are able to inhibit the pro-inflammatory cytokines or enhance the ability of the anti-inflammatory cytokines or say host responses, you will probably come up with the strategy to deal with mycobacteria, the host-directed therapy. Even the phagolysosomal fusion, because mycobacteria prevents it. So if you are able to promote that, mycobacteria will be eventually cleared. You don't really need antimycobacterial drugs, and that's where the ZMP1 is a target. I'm not really talking about all of these targets because there are so many of them. I'll be only focusing on 2 or 3 of them. Coming on the right hand side, you have got Mycobacterium tuberculosis. So many of the drugs, they act by inhibiting the cell wall biosynthesis. Although people wouldn't want to develop drugs which will act against cell wall biosynthesis, like mycobacterial cell wall biosynthesis, because the chances of developing resistance are high. But still, there are few targets in this area. Then there are other targets which disturb the redox homeostasis in mycobacteria. Meaning, I told you about mycobacteria has devised strategies to survive inside the macrophages, alveolar macrophages, inside the phagosome, and then redox survival is very critical. So if you are able to inhibit enzymes like MTR or MSHC, which are responsible for producing intermediates which eventually protect the mycobacteria from the reactive oxygen species or reactive nitrogen species, you'll be able to kill mycobacteria. And then there are other targets, including the ones I told you about, DNA dependent RNA polymerase. Then you have got gyrase A, gyrase B, and then there are other enzymes which are responsible for DNA replication and protein synthesis, which happens in the dividing mycobacteria or replicating mycobacteria. But then if the mycobacteria is dormant, probably it's not replicating, it is under hypometabolic stage, these drugs probably wouldn't be effective. But then people are still trying to develop drugs because with the hope that these will kill the replicating mycobacteria. And there are other targets, including ATPE, which is the target of beta-quilin, which was introduced in 2012, which basically interfere with the oxidative phosphorylation. And so there are the point is there are various host-directed therapies and there are various pathogen-directed therapies which can potentially be useful for treating the multi-drug resistant TB. So coming back to host-directed therapeutics, a closer look at that. So these therapies are intended to increase the success of TB treatment by immunomodulation and or immune augmentation. Right? So depending on what strategy you use, you can either increase the immune system function, or you can selectively tame down the immune system so that the underlying inflammation is controlled and the tissue damage is prevented. So given the complexity of the disease progression, a single immunotherapeutic is unlikely to be effective in the full TB spectrum. So you have to have a therapeutic which will act in unison with other anti-therapeutic or anti-TB drugs. And that's where the emergence of the concept of precision medicine approaches have come up. It's because you have because every TB patient is different. Like some patients may suffer from diabetes, some patient may suffer from HIV, or some patient may be just lone TB thing, like so suffering from only TB. So not every approach which is like host directed will work in all patients. So you have to have a personalized approach. And I told you about the synergism with the current anti-TB drugs is very important, so that you try and take care of the not only the drug susceptible TB as well as but also the drug resistant TB, because eventually if you're using the host directed therapeutics in synergism with in synergy with the current anti-tubercular drugs, you probably will be able to improve the long-term outcome and reduce the duration of the therapy, and the disease is probably completely eradicated from that patient. There are four strategies. The first one is to modulate the pro-inflammatory mediator to dampen inflammation. I told you, because it's basically the immune system failure to clear the TB infection which causes TB. So if you are somehow able to enhance immune system efficacy, you essentially will be able to get the desired outcome. The third strategy is enhance bactericidal mechanisms in macrophages, so that the MTB killing happens and the bacilli growth is reduced. Right, so that's another important thing. The next one is disrupt structure and penetrate granuloma to expose MTB to anti-TB treatment. So that's where the whole premise lies, because the currently used anti-tubercular drugs are not able to penetrate the granulomatous tissue, which is a complex environment. They are not able to clear the infection, and the mycobacteria resides in the in that particular state, in the dormant stage, for a very long time, and eventually not able to clear the infection is not clear. So you have to have a host directed therapeutics which will kill, which will disrupt this granulomatous structure and bring the mycobacteria out, so that the anti-tubercular drugs are able to kill it, right? So coming back to pathogen directed therapeutics, so if you take a holistic view of the available drug structures or target structures for killing mycobacteria, that is pathogen directed therapeutics. So MTB genome contains approximately 4.4 million base pairs, out of which actually encode greater than 4,000 genes. Out of these 4,000 genes, like these 4,000 genes essentially are responsible for expression of more than 500 essential proteins. And out of these 500 essential proteins, only less than 10 are used by currently available anti-tubercular drugs. So you see there is a huge technology gap in this area. Now, out of these 500 essential proteins, only 73 are, I would say, single proteins. All others are part of some or the other complex, a multi-domain or multi-protein complex. So that's where you have got like 63 single protein targets which can be exploited for drug discovery and development, and that's where I would say most of the efforts which are going on in the globe, around the globe, about anti-tubercular drug development. So people have tried to focus on these 63 targets in some or the other way. Some are really at the nascent stage, like assay development and stuff like that, whereas some have advanced to clinical development, maybe say phase 1 or phase 2 clinical trials, right? So this is again the holistic view of what all MTB targets are used by the currently available drugs, including the first line as well as second line drugs. Isoniazid, it inhibits the INHA. The D-cycloserine, which is another second line drug, which also is responsible for inhibiting the cell wall biosynthesis. You have got ethambutol, which acts on EMB. AB, bedaquiline, it inhibits ATP synthase. You've got rifampicin, which inhibits the RNA-dependent, DNA-dependent RNA polymerase. You have streptomycin, kanamycin, amikacin, capreomycin, which inhibit protein synthesis by targeting the ribosomes, bacterial ribosomes. You have got fluoroquinolones, which inhibit DNA gyrase, which is responsible for unwinding or uncoiling of DNA. You also have got pyrazinamide, which in a way acts on FAST1 target. So this is the whole summary of what all presently available anti-tubercular drugs. So ideally, you probably would want drugs which do not act against these targets and may act on some other targets, because otherwise there is unless you have a reason to believe that a drug which is acting on the old target is acting by a novel mechanism of action, you shouldn't be touching the old targets because there is a very high propensity to develop cross resistance. Now coming back to new approved drugs, like I told you, there has been a huge discovery gap, huge technology gap as far as the new drug discovery for TB is concerned. But recently, like in this decade or so, 3 drugs have been introduced. First one being bedaquiline, which was introduced in 2012, which acts on mycobacterial ATP synthesis. It inhibits the mycobacterial ATP synthase, and that's how it depletes the mycobacteria of ATP, and it cannot really replicate and survive. The second one is delamine, which inhibits the mycobacterial cell wall biosynthesis, and eventually it acts as antimycobacterial or anti-TB drugs. The latest one, which was introduced last year, pritomanine, which is actually a poison for bacterial respiration under anaerobic conditions. So this is particularly useful when the mycobacteria is in dormant stage, where it is in hypometabolic stage inside the granulomatous tissue, which is not accessible by other drugs. So pritomanide, which is again a prodrug, the nitro group is responsible for its activity, in the sense it gets the nitro group is reduced, and the benzimidazole gets converted into reactive metabolites, and it goes and binds the respiratory, like the cytochrome, I think it's cytochrome BC1, it gets inhibited, and eventually you see the antitubercular effect, right? So these are the 3 drugs which were introduced in this decade for TB treatment. So coming back to the next part of pathogen-directed therapeutics, most of the advanced TB drugs which are in phase 1, 2, or 3 trials, they target energy metabolism in mycobacterial cell, particularly the ATP synthesis, as well as the respiratory chain cytochrome BC1 complex, and some of them they target cell wall biosynthesis. Several molecules are under various phases of development, including biological testing, preclinical, clinical, phase 1, 2, or 3. There is a working group on TB drugs. This is the website. You should go and check there frequently. You can actually join this consortium as well. There is a pipeline section under this. They update about the current TB trials, including the known as well as the novel drugs. They update every now and then about the latest happenings on TB drugs. So they're working relentlessly to accelerate discovery and development of new drugs for the treatment of tuberculosis by bringing together all the stakeholders, including physicians, clinicians, drug discovery researchers, as well as the patients, right? So including the biologists which are working on TB. So it's kind of an open forum where people can openly discuss ideas, get together, and in a way progress to the clinical development. In fact, whatever is there in phase 3 development, they are all by the working group on TB group, by this particular alliance. And then various stakeholders contribute in different ways, including money, the efforts, and even the clinical trial efforts for that matter, apart from the drug discovery efforts. And that's how the TB drug is advancing. Because like I said, TB is a disease of developing or underdeveloped nations. So big pharma companies would probably be not interested. I mean, they were never interested. And that's why this consortia, which is supported by WHO, which is supported by some humanitarian organizations like Bill and Melinda Gates Foundation, Glaxo Wellcome Trust, and many others, so that they can continue working on these diseases, which eventually haunt us, the developing as well as underdeveloped nations. South Africa has the largest number of TB patients per 100,000 patients. So you can imagine the African nations, most of which are underdeveloped, eventually suffer from TB, and AIDS also is very prevalent. So that's why TB is a major concern from them. So this is where we are coming towards the conclusion, but the most important part of the presentation. So out of many targets, like I told you, people are working on this 63 single proteins as antimicrobial targets, some of which are essential, some of which are related to virulence. So then some of these are summarized here. Three of them actually. The first one is DPR-E1. It's as a novel anti-TB target. Now, if you search the literature, it's not new. In this sense, people started working on DPR-1 as early as like early 2000. But then this probably was identified after the after the mycobacterial genome was sequenced, and the people identified that DPR-1 is an important target, which essentially is a highly vulnerable and fully validated tuberculosis drug target. Whenever you select a drug target, you have to make sure that there is no similar target exists in the host. Say something like DHFR, dihydrofolate reductase. So if you are working on mycobacterial dihydrofolate reductase, you have to make sure that it does not, the molecules which you develop for TB treatment, the MTB DHFR inhibitors, they should not inhibit the host DHFR, otherwise there will be precipitation of toxicity. So that's where the validation comes into picture. So you only work on targets which are validated, fully validated, and mycobacteria is highly vulnerable, meaning if you inhibit those targets, mycobacteria will die, or its virulence will be compromised. Because there are alternate pathways, then the mycobacteria eventually will survive even if you inhibit the target with very high potency. Right. So about DPR-E1, it's an FAD containing oxidoreductase. If you see the enzyme commission code, it's class 1. It's an oxidoreductase. It catalyzes the first step of the two-step epimerization of conversion of DPR to DPA. The process is shown here. So it's a two-step process. If you look at the mechanism, so the second hydroxyl group gets converted to ketone, and then it gets epimerized. So from alpha it gets beta, and this essentially is responsible, or this is DPA is a precursor for mycobacterial cell wall arabinine or arabinine synthesis, meaning if you inhibit DPR-E1, mycobacterial cell wall synthesis will be inhibited, right? And that's where that's why it is a vulnerable target. So if you are inhibiting mycobacterial cell wall synthesis, eventually there will be higher permeability of the mycobacterial cell wall to, there will be faulty cell wall biosynthesis, and drugs can now easily penetrate through highly lipophilic barrier, which is made up of mycolic acids, which is not permeable to majority of the drugs. So if you are inhibiting the cell wall biosynthesis, the conventional anti-tubercular drugs will eventually act nicely, and they'll kill the mycobacteria. So in a way, DPR-E1 is a target which these inhibitors can work in synergy with the known anti-TB drugs. A lot of compounds are reported in the literature, particularly the compounds from the benzothiazidone series. They're like nanomolar inhibitors, like 1 nanogram per ml or something. So very, very potent inhibitors, and these are the initial. BTZ-043 was one of the earliest compounds. Now, if you look at the middle structure, CT325 and CT319, the only difference between, in case of CT325, there is a nitroso group. In case of CT319, it's a nitro group. The basic difference between these two drugs or these two molecules is both are DPR-E1 inhibitor. CT325 is a covalent inhibitor, so it modifies, it gets covalently attached to one of the cysteine 387 in DPR-E1 of mycobacteria, whereas the same CT319 with a nitro group, it binds to the same target, but it does not covalently modify DPR-E1. So that essentially means the covalent inhibition of DPR-E1 is not really needed. So you can have non-covalent inhibitors. You can still inhibit the DPR-E1, but then when it comes to nitro compounds, they are highly toxic, but because as long as they are doing their job of killing mycobacteria, you probably can still take them ahead. Otherwise, any drug discovery researcher would hate to see nitro-so and nitro groups in the molecule. You all are aware of NDMA impurity in ranitidine as well as famotidine, which led to recall of ranitidine batches. So it is actually a nitroso containing impurity, because these impurities are highly mutagenic, and so are these compounds containing nitroso or nitro compound. Nimesulide contains nitro group. Nimesulide is a banned drug outside India, but in India we still use nimesulide. But then that's a different story. But nonetheless, it's a very toxic drug. The point is, nitro-containing groups or compounds are highly toxic, and one should not really think about that. But irrespective of that, because these were the earlier DPR-E1 inhibitors, eventually people started use these compounds for the so-called structure-based drug design, and eventually developed safer compounds which were devoid of nitro. Now, this is a picture of a three-dimensional structure of DPR-E1, which is, there are many structures of DPR-E1 in protein data bank. You can, if you just search with the name DPR-E1, you will see a lot of structures with the ligand bound in the binding cavity. And this is the structure of CT319, which acts by non-covalent inhibition of DPR-E1. The particular molecule, that the particular residue, the cysteine 387, is somewhere here, very close to the nitro group, but this compound does not inhibit. So if you see, these angular structures are water molecule, crystallographically used crystallographically observed water molecules, and these dotted white lines, they are the hydrogen bonds. So if you see the hydrogen bond between this nitro group and the backbone carbonyl oxygen of the DPR-E1, so the more the number of these interactions, including hydrogen bonds or hydrophobic interactions, better is the binding with the target, and eventually better is the potency. These molecules are highly potent molecules, nanomolar inhibitors. So they bind very strongly to the DPR-E1. And all of these things you can study with the help of molecular docking. Like the other day, I was talking with Dr. Kumar, and we were discussing about molecular docking. So when it comes to molecular docking, if the active site is big enough, you dock any and every molecule, it will probably go and bind, because whatever we are trying to understand or simulate what happens at the biological level with the help of computer, it is an oversimplification what happens at the biological level in vivo conditions, right? So when you are using molecular docking as a tool to understand, as a tool for developing or designing new molecules, you should be cautious enough to make sure that you understand the limitations of the techniques which you are using, and molecular docking is no exception to that. So you shouldn't be overexcited just to see that the docking score is super great, but you should also understand the likelihood of there are other techniques, additional techniques, which can confirm whether the compound is likely to be binding to the molecule, to the target, or not. Like conducting molecular dynamics simulations, and see how stable is the interaction between the inhibitor, the design molecule, your molecule, and the target. So there are ways to study that. But then whenever you use molecular docking, you should be cautious. And there are various free softwares like Autodock available, and it's an excellent software. You just need to understand how to use it. There are many YouTube videos available, and eventually you can be a pro in using Autodock for whatever purpose you're using it for. So this is a summary. This is actually a picture from one of the JMC perspective, which was published in 2018 on DPR-E1 inhibitor. So you see many of these molecules, they do not have nitro compound. If you see benzothiazinones, there is a nitro group present, but most of them, they do not have nitro group. So they have used the molecular modeling tools to come up with molecules which are less toxic, which are more potent, and eventually do the job of killing the microbacteria by inhibiting DPR-E1. And the structure because a lot of structures are available, people have used extensively the structure-based approaches for designing DPR-E1 inhibitors, right? This is another target which I have been actively working on for quite some time. The name of the target is MTB-GUA-B2, and it's again a validated target. The guy whom I work with in South Africa, Dr. Vinayak Singh, he has developed assays, or rather, he has worked on this target. He's an active researcher in this area, and they've got a molecule which is in, I think, it's phase 1 trials or so, or probably advanced to phase 2, which acts on MTB-GUA-B2. Other name for MTB-GUA-B2 is MTB-IMPDH, which stands for uh, which stands for Mycobacterium tuberculosis inosine 5-monophosphate dehydrogenase. It's an NAD plus containing oxidoreductase. Again, this is an oxidoreductase which characterizes the penultimate step in the de novo guanine nucleotide biosynthesis, that is conversion of MP to XMP. The process is shown here. So now IMP gets covalently attached to this enzyme, and eventually in a series of electron transfer reactions, it gets converted to XMP. So if you see, it's an oxidation reaction. The hydrogen in IMP gets converted to oxygen in XMP. Now this XMP eventually gets converted to GMP with the help of GMP synthase, and it gets converted to the nucleotide version. Now, this is the branch from IMP. One wing goes to, or one branch goes to GMP. The other branch goes to the AMP, right? So this is an essential reaction in the de novo guanine nucleotide biosynthesis, which is required for DNA synthesis. There is another pathway, which is salvage pathway, which uses preformed nucleotides for forming the guanine nucleotides, as well as the adenine nucleotides. But then if you compare the salvage pathway and the de novo biosynthesis pathway in mycobacteria, particularly in the rapidly dividing mycobacteria, it is mostly dependent on the de novo biosynthesis pathway. And people have shown, the researchers have shown that the salvage pathway, even though there is excess of preformed nucleotides, it does not reverse the inhibition by IMP or MTB-GUA-B2 inhibitors, right? Meaning the mycobacteria is still killed. So what I'm trying to tell you is MTB-GUA-B2 is a genetically validated, vulnerable and a bactericidal drug target, and many people are working, including my group, on this particular target. So coming back to MTB-GUA-B2 inhibitors, so this is a paper from Vinayak's group where they have identified the isoquinoline-based inhibitors, which were very potent inhibitors. Now again, if you see, there are the middle compound number 21. There are 2 nitro groups, but eventually they got rid of nitro group because they're so toxic. So if you look at compound number 47, it's MTB-IMPDH isophthase 35 nanomolar. It's MTB MIC90, which is the concentration required to kill 90% of the mycobacteria. That is 3.1 micromolar, and then compound 1, resistant mutant. So they have also developed a resistant mutant from the original hit, that is compound number 1. Its IC50 is greater than 100. That means it is essentially acting at the same site. So this is a way to prevent, this is a way to demonstrate whether there will be development of cross resistance for the new compounds or not. And then on-target selectivity, there is an on-target selectivity, meaning it specifically inhibits MTB-GUA-B2 and thereby kills it, right? It does not really act by any other mechanism, right? So as far as the structure-based studies are concerned, Vinayak's group, they have come up with the, they have deposited crystal structures of MTB-GUA-B2. There are many crystal structure. Now, if you see the figured lesion, it says MTH, so it's not a typo. It is MTH, that is Mycobacterium thermophilus, which is a closely related organism, which was used for Like the MTH GuaB2 was used for crystallization because the MTB GuaB2 wouldn't work as far as crystallization for X-ray crystallography is concerned. So they came up with MTH, but then they have genetically modified it in such a way that the active sites of MTH GuaB2 is very similar to MTB GuaB2. So in a way, you are mimicking whatever is there in the MTB GuaB2. So if you see here, this particular on the right hand side, the ball and stick model is for the inhibitor, one of the inhibitors from the isoquinoline series. Whereas on this side, you have got the IMP, which is the substrate, which is bound in the target. So you have got a substrate binding site, that is IMP binding site, and you have got a cofactor binding site, and that cofactor is NAD plus. So we are targeting cofactor binding site for developing inhibitors. Now, if I tell you about COVID-19 or the SARS-CoV-2, ribavirin, which is also they have demonstrated that ribavirin is a potent SARS-CoV-2 inhibitor. It acts by inhibiting human IMPDH. It acts by inhibiting human IMPDH, which is similar to MTB GuaB2 in MTB. So what happens is ribavirin binds in the substrate binding site, that is IMP binding site, whereas all other inhibitors which we have worked on or which we work on essentially bind in the NAD binding pocket. So with the help of the structure-based studies, the three-dimensional analysis of the crystal structures of the enzyme, the target, you are able to design compounds which will increase the inhibition potential or potency of that particular drug. So I was talking about the NAD or the substrate binding site. The cofactor binding site. So it basically, this is the structure of NAD, and it basically is it has got three pockets. One is N subunit, where the nucleotide part binds, the sugar part binds of the NAD part, the nicotinamide part. Then you have got the P subunit, where the diphosphate of NAD plus binds, and you also have got the adenine binding part. So if you remember, I told you just a few minutes ago that if you are trying to inhibit a target whose counterpart exists in the host, you should be careful. But here, although human IMPDH is there, but there are substantial differences between human IMPDH and MTB GuaB2. And that's where we have tried to utilize those differences for designing the pathogen selective inhibitors. Now, if you see here in this particular, this is from a crystal structure analysis of MTB GuaB2 and human IMPDH2 structure. So we have the cofactor binding sites are aligned onto each other. So if you see, the green one is where the mycobacterial cofactor, that is NAD plus, will bind. So it's actually the nicotinamide part binds at the same place, but the adenine part, the AE or A beta subunit, that's very different. And the way the cofactor binds to the MTB GuaB2 as well as the human IMPDH, because there are inherent differences, and these differences are utilized for designing of selective MTB GuaB2 inhibitors. And we have done extensive work on the structure-based analysis or structure-based design part of the MTB GuaB2 inhibitors. And we have got a couple of series which are, I wouldn't say very potent, but moderately potent. And this is where our contribution is. We published this work 2 years ago in Bioorganic Medicinal Chemistry later. So these are the two molecules from the initial series, which eventually were very quite potent, I would say, 3 micromolar at the target, the MTB GuaB2 inhibition potential, but their MIC90 was quite high. Obviously, they had the tissue penetration issues. They were not bad, but then 100 is not something what you would expect. And we have also proved that these two compounds, these two initial lead compounds, they were active in the target specific assays. And that's where in a way we were very much convinced that these compounds were true inhibitors of MTB GuaB2, and further work with Vinayak is in progress. So basically we design and synthesize molecules, and Vinayak helps us to test the anti-MTB activity. And his collaborator in Italy, Dr. Menico Rizzi, he is basically responsible for the MTB GuaB2 enzyme inhibition study. The For the selectivity part, the human IMPDH inhibition study, we do it in our lab. And for the MTB, because it needs BSL-3 facility, we send it to Menico as well as to Vinayak for testing, for biological testing. Right? This is the last part of the presentation, wherein another target, MSHC, which is again a novel anti-TB target. I'm going to talk about this. I think a couple of more slides and we will be done with the presentation. So a few things about MSHC. It's an essential enzyme for the in vitro as well as in vivo growth of M. tuberculosis. Now the idea is very simple. If you remember, or rather if you know, we have glutathione in every single cell of our body, which acts as a cellular antioxidant. Whenever there is attack by free radicals or other harmful species, the glutathione will try and neutralize that attack by either covalently coupling to the free radical or neutralizing free radicals or by other mechanisms. So similarly, for mycobacteria, MSH and EGT, these are the two, I would say, the mycobacteria. MSH stands for mycothiol, which acts in a similar way. Glutathione works for our cells. So MSH and EGT eventually inhibit the reactive oxygen as well as reactive nitrogen species, which are produced by the attack by anti-TB drugs, oxidative stress, nitrosative stress, or respiratory burst, which is an immune mechanism for killing the mycobacteria or for killing any pathogen for that matter. So MSH it protects MTB from DNA damage, lipid peroxidation, from anti-TB drugs. So in a way, it's a survival strategy for MTB. So these two guys help MTB in maintenance of redox balance and survival in macrophages and hosts. So for a very long time, they will survive very nicely, under hypometabolic states, right? And that's where somebody came up with the idea that if you are able to deplete mycobacteria of MSH, so probably it will be susceptible to the killing by all other harmful stimuli like anti-tubercular drugs, oxidative stress, nitrosative stress, as well as respiratory burst. And that's where people came up with this idea of developing MSHC inhibitors, which are, now this is the reaction which is catalyzed by MSHC. If you see, this is a disaccharide to which a cysteine is added. In case of glutathione, it's a tripeptide. Cysteine is one terminal amino acid, and apart from that, you also have two amino acids. But in mycobacteria, it is the sugar units instead of the peptide unit. But ultimately, it is the cysteine part which is responsible for all the beneficial effects of MSH. So MSHC catalyzes this particular reaction. Now, once the cysteine is added, there are additional reactions, one or two reactions down the line, which will convert this intermediate into MSH, and that MSH will do all the beneficial jobs for mycobacteria. So the idea is, if you inhibit MSHC, eventually you will, you will inhibit formation of MSH, and eventually the mycobacteria will be more susceptible to killing by other stimuli. And people came long ago, people came up with inhibitors belonging to the MSHC category. This is one of the earlier prototype inhibitors, which looks like benzodiazepines or some tricyclic antidepressant thing, but it does work. It's quite potent MSHC inhibitor, and there are efforts underway which eventually have demonstrated the utility of this approach. So just like other targets, people actually came up with the three-dimensional structure of MSHC, and eventually, tried to study how MSHC inhibitors would eventually bind to it and cause the inhibition of MSH biosynthesis. Right. So again, with the help of now, if you see here, it's a metalloenzyme. On the right hand side, you see a zinc here. So all of these studies have led to identification of quite potent MSHC inhibitors, right? So the whole idea is to study single proteins, eventually make sure that you develop potent inhibitors. Before that, you will have to develop the assays to quantify or to screen the library so as to identify the potent inhibitors, and you then in a typical lead identification and lead optimization phase, you take it to the next level. Once you identify the lead candidate, you carry out the efficacy studies in vivo models, and then all the preclinical safety pharmacology and then toxicity studies along with the pharmacokinetic analysis, and make sure that your compound is safe to be used in humans, and then you file an IND. It goes to the phase 1 trials, then from there phase 1 to phase 2 to phase 3, and then you get approval. Of course, it's a long process, but unless you start today, you will not be able to come up with a drug which will be introduced in the market, say down the line 10 years. So again, you have to keep that thing in mind, whether this drug will be useful after 10 years, right? So that's where your initial hypothesis has to be very, very strong, right? If you come up with a target which has got a substitute in mycobacteria, then it's of no use. Substitute in the sense an alternate pathway which will, like, you inhibit the target, deplete mycobacteria of some essential metabolite. But if mycobacteria is able to make that metabolite by a different mechanism, by an alternate pathway, then your inhibition of your target, initial target, is of no use. So a lot of thought process goes behind all of this, and eventually people come up with ideas. But then, like I said, because the major pharma companies are not really working in this area, so the drug discovery is happening with the help from philanthropic organization, but it is still slow. But unless you have got some lead compounds, some interesting ideas, there is no way to move forward in the sense that there is no further drug development, and people are working in silos as far as the anti-MTB therapy is concerned. So a lot of people need to work on this in unison, and then of course, because it's a national health emergency, we have to be serious, and government is serious about that for sure. I mean, they've got a very ambitious plan of eradicating TB from India by 2025, which seems impossible, but unless everyone works towards this common goal, we are not going to achieve that particular thing. So the last slide, what is the take-home message? So the first very optimistic point is there are unprecedented opportunities for anti-TB drug development. A lot of literature available. Pathogen-directed therapy has got a high probability of resistance development, which is true. If you are putting a selective pressure on the mycobacteria by inhibiting its essential proteins, it will come out with a strategy which will make the drugs ineffective. Host-directed therapies, because you're not putting selective pressure on the mycobacteria, they've got a great potential. But then you have to have, your approach for host-directed therapies has to be very concrete, and it is this area where a lot of drug repurposing is happening. Just like you see a lot of drugs being repurposed, anti-inflammatory drugs being repurposed for COVID-19, similar approach can be done. So if you are an aspirant in anti-TB drug discovery, the best way to start is with the host-directed therapies, where you start with drug repurposing and then eventually come up with very solid and concrete strategy. Because you are dealing with known drugs, the probability of success is very high, and eventually you can achieve what you dreamed of. And the last one is structure-based approaches are quintessential for the discovery of host and or pathogen-directed therapy. So there is no substitute for, of course, there are ligand-based approaches available, but then structure-based approaches make life much, much easier. Although I would say I'm more biased towards ligand-based approaches, because at times they're not dependent on the availability of the three-dimensional structure. So, but then each approach has its own advantage or own capability and own limitations. Anything you do, as long as you are able to come up with very interesting lead compounds or hit compounds, that is perfect, because ultimately that is necessary. So that's all from my side. With folded hands, stay safe, maintain social distancing, and do whatever you are doing safely. And if you have got any further questions, I would love to answer those questions. Thank you so much.
Yeah, thank you, sir. We got an interesting and novel research knowledge regarding all the aspects of Mycobacterium tuberculosis. We are enriched by the exposure to various novel targets that can lead to the development of a new molecule in future. Thank you again. Now coming to the question and answer session, I request any participants from Zoom can unmute and ask the question, and those who are in YouTube hot chat, they can send the question in the chat box. I will take your questions. Thank you.
I mean, I know it is a complicated area, but please feel free to ask.
Hello, sir.
Yeah.
Good afternoon.
Yes.
Sir.
Good
Sir.
afternoon.
Hello.
Yes, I'm
First of all,
listening.
thanks for the thanks for the presentation, sir. I am Naveen from MPharm Pharma Chemistry. Sir, I have a doubt, sir. Why can't we expect drug resistance before drug designing?
Well, very interesting question. See, what happens is usually many times when it comes to antimicrobial drug resistance, or rather the drug resistance in mycobacteria, which makes the therapy ineffective or inefficacious. At times, say particularly in case of TB, what happens is patients do not stick to the therapeutic regimen, and the mycobacteria are exposed to the sub-therapeutic concentrations of the drugs, and that's where they devise the strategies to come up with the resistance. Now, in fact, regarding your question, people in fact try and develop the drug resistant strains right away while the drug is still under development, so that you come up with strategies or molecules to overcome the drug resistance, because drug resistance is going to happen no matter what eventually. But then if you are ready with, it's like, after this India-China thing at the border, we are all on the vigil, right? The Air Force is on the vigil, everyone is on the vigil. The soldiers have gone there, 250,000 soldiers. So this is the same strategy. In fact, in our own work, or rather Vinayak's work, I would say, they have, right in the beginning, they have come up with a molecule. They have identified the drug resistant strain and the changes what happen in the target, and whether the compound becomes in a way ineffective or not. So they have developed those strains by exposing the compounds to sub-therapeutic concentration or suboptimal concentration. The mycobacteria eventually will lead to changed or target modification, and then they isolate that particular protein and use it for in vitro study. So if you have a compound which eventually is resistant to one particular drug, say original molecule, and then eventually you develop molecules, if it also becomes resistant, if it also shows resistance, if the target shows resistance to the target, maybe in the form of very low potency or something, then you in a way can prove that the compound is binding at the same site. But it is also possible that if you come up with a new structure, it may bind to the same target, but at a different site. So in that case, you will see that even though the drug is, even though the target is resistant to say drug, the original drug, but it is not, it is still showing inhibition for the second molecule, which is maybe structurally similar, maybe structurally different. It is possible that the compound is occupying a site which is different than the original drug, right? So people do that because that is just a strategy to be able to prepare yourself for the eventuality and develop molecules which eventually will be useful for the resistant or the mutated version. And in fact, kinase inhibitors, if you see, in case of kinase inhibitors, you eventually have first generation kinase inhibitors. So the kinases become resistant to that. They've come to second generation, third generation, fourth generation, like that. So it's a known phenomenon, and people are ready. Great question, I must say that. But people do try to develop the resistant strains right away.
Thank you, sir. And I have another doubt, sir.
Yes.
Are we using any kind of therapies like stem cell therapy or gene therapy in this tuberculosis diseases?
I don't think stem cell therapies, but then when it comes to say host-directed therapies, people are using a lot of different therapies which basically are based on some polypeptide molecules. But I don't think I have read anything about stem cell therapy for tuberculosis for sure. I don't think so.
For improve the immunity of person for
Yeah.
using stem cell therapy.
Well, I'm not sure. At least I haven't read, but I'm sure there would be somebody thinking on those lines. So it's like imparting, like introducing some genes, like introducing some genes which will increase the overexpression of a particular type of cell, maybe Th1 cells or something. In fact, as far as immunotherapy goes, again, a great question, I must say. One of the approach of host-directed therapies is harnessing the potential of cytotoxic T cells to kill the pathogen cells, because the cells will express, the mycobacterial cells will express antigen, and these antigens like eventually will have, say, programmed death receptor 1, PD1 receptor, and then there will be equivalent ligand, PDL1 ligand. So PD1 PDL1 interaction will make or will in a way induce the production of cytotoxic T cells, or the T cells will be primed in a way to kill the microbe. They use this PD1 PDL1 axis as a very prominent therapeutics for cancer immunotherapy. So what you're trying to do is you're telling the T cell that there is some external invasion which has happened, you go and kill that particular invasion. And that's how the PD1 PDL1 inhibitors, they have got shown, most of those are antibodies. They have shown a very great potential in eliminating cancers, certain types of cancer, not every cancer. So you are making body stronger, or rather immune system stronger, in eliminating the threat. And exactly similar thing is also being done in case of TB or infectious diseases for that matter. But I wouldn't say the compounds have really advanced to a higher, like advanced clinical stages. So some of them are in preclinical stages, but people are thinking on those lines. Yes. Again, a great
Yes.
question. Yes. Thank you so much.
Thank you, sir. Thank you,
Yeah.
sir.
Sir, I have a question, sir. I'm
Yes,
from
please.
the fifth year.
Yes.
Myself, Rupa Pravalika. I would like to know why rifampicin resistant, even though it is less common, but deaths associated with it are more. Why? When compared
Essentially, can you repeat your question, please?
rifampicin resistance, even though it is less common, but deaths associated with it are more when compared to extended resistance patterns and multi-drug resistance patterns.
Yeah. So usually what happens is, if you see now, rifampicin is used throughout. So initially, if you look at the DOTS therapy, they start like first 2 months are 4 drugs peri, that is pyrazinamide, ethambutol, rifampicin, and isoniazid. Right. So after 2 months, only rifampicin and isoniazid, they essentially are kept for up to 9 months. Right now, as far, because rifampicin is used so very widely, rifampicin resistance is definitely, I mean, it does happen. But then apparently, like your question, why, I mean, it is less common, but still the deaths happen. I mean, again, I would say the host associated toxicities, say particularly the INH as well as rifampicin. I mean, at any point of time, if you see, rifampicin will never be given alone. It will always be given with INH. And if you look at rifampicin, it has got other issues as well. Apart from hepatotoxicity, it is also toxic to the rapidly dividing cells. I think it's a myelotoxicity or hematopoietic stem cell toxicity is also rampant. Right. So eventually the resistance develops, and because you can't give, so basically it's all a common, I would say, common effect. And moreover, once the drugs, once the mycobacteria develops resistance, so the drugs are not effective, and eventually the active progression also starts. But again, if you ask me, every patient is different for that matter. Say presence of comorbidity, his or her habits, alcohol use, smoking, and even the environment, which affects us in a greater way. So if you are, if that person is from lower middle class, then the kind of nutrition he or she will be eating will be very different. So all these factors will come together, and the end result, the worst outcome is seen. But I would say, I don't know if I have given the answer to your satisfaction, but it's complicated. As in, It is very complicated. So, but basically the toxicities associated with the chronic use of very high doses of drugs that is responsible for all the issues actually.
Thank you, sir. And one more question, sir.
Yes,
Uh,
please.
in case of HIV infection, even
Mhm.
after use of antiviral therapy and also the increase in the CD4 cell count,
Yeah.
there are more chance of getting TB opportunistic as opportunistic infection. Why?
Yes. Well, again, if you look at the antiretroviral therapeutics, of course, the HIV is, HIV is marked by the lymphocyte count decrease, and even if you increase, in fact, I was reading yesterday itself, people have tried to use this approach. It's like because the lymphocytes are less, so eventually you end up you fail, like the body of that person fails in there is still I would say technology gap in terms of the LTBI, that is HIV TB co-infection, and what we know about what antiretrovirals do. In fact, then UK will come, and early career researchers from India will also take part in this. And along the 4 days, we would be discussing on this particular aspect, like the HIV TB co-infection, and what are the strategies to mitigate the risk associated with, say, TB, because every fourth person in India dies of who is suffering from HIV dies of TB. Right. Again, I would say there is no definitive answer to your question. People are still working on that, as in still trying to find answer, because TB was never never a mainstream research or rather disease of the researchers. So people have now in a way come to senses, and they're trying to come up with strategies. In fact, if you ask me about the vaccination strategies, particularly, I mean, BCG works with limited efficacy, but people have also come up with additional mycobacterial proteins, which essentially are responsible for the induction of immune response against tubercle bacilli. But those vaccination approaches are also not very effective. So in a way, what I'm trying to tell you is mycobacteria is a stronger version of what we see today. And it is trying to, or it has already devised strategies to thinking that eventually we won't be able to counter them. Meaning, even if we devise something to counter that, it is still not we still are not successful. So there is a long technology gap between what we know and where we have to go, and what mycobacteria is capable of. So there is again no definitive answer to your question, but what you said exactly is, I mean, that's very true. Even if you increase the lymphocyte count, it still doesn't do. In fact, it is a failure of the immune system which leads to TB. And obviously, in case of HIV patient, even if the person is on antiretroviral therapy, it is still the immune system in healthy individuals also fails. So leave aside the HIV patient, which is already immune compromise, like the compromised immune system. So it is I would say it is still not really known with certainty as far as the answer to your question is concerned. People are working, a lot of research is going on. If you see the number of articles published in PubMed in last like 5 years, there are huge articles, the huge number of articles have been published, most of them featuring on the molecular aspects related to diagnosis and treatment. Right? Although drug discovery has always been there, particularly in last 10 years or so, but then the molecular based approaches, there are inherent difficulties in when it comes to mycobacteria, because it's a slow growing organism for sure. So that's one thing. Apart from that, it is very powerful, right? And it has acquired all the necessary mutations already to counteract whatever we can even think of. But then that shouldn't be, that shouldn't demoralize us from working on MTB, because eventually we'll have to, I mean, it is already a grave situation, although we fail to recognize it. Now that there is COVID-19, there was a point where there were more number of patients who would die of TB than COVID-19, but it just surpassed those numbers. So if you take similar precaution in case of TB, believe me, TB spread would have been controlled a long time ago. Like people wearing masks and preventing the disease from being spread up while you are still in the public places. The best part is the virulence of MTB as far as its communicability is concerned, or its communicable potential is concerned, is still low. Right, we can an infected person with active pulmonary TB can only infect 8 to 10 patients. Right, although I mean one-fourth of the world population already harbors latent TB, so it's I mean there is already enough reservoir of patients, TB patients already. But then some institutes like the one in Chennai, the National Institutes on Tuberculosis, they are doing extremely good work, although it is still a long time before we have some concrete outcome of their results. But then the genomic studies featuring on the capabilities acquired by Mycobacterium tuberculosis, they are still poorly understood. That's exactly why we are not in a condition to counteract those strategies adopted by MTB and come up with very powerful anti-TB approach. I wish I could have more specific answer to your question.
Okay, sir. Thank you, sir. Can novel drug therapy ensure that it is free from resistance?
Well, like I said, eventually it will, like these drugs will develop resistance just like any other antimicrobial. But again, there is a difference. If you have got a pathogen-directed therapy, which is not targeting the essential proteins, like if the therapy targets the virulence factors or something like that, then we are putting less selective pressure on the mycobacteria, and the development of resistance will happen after a very long time, as compared to the inhibition of essential targets of mycobacteria. And mycobacteria will eventually find out ways to come up the inhibition around the inhibition by the powerful drugs. So that's exactly, in fact, if you read about bedaquiline, Indian government has restricted the use. In fact, it was prevented from using in patients with active TB with the same fear that it will develop resistance very soon. And believe me, it's the thanks to all the physicians and everyone whose indiscriminate prescribing of antimicrobials, including anti-TB drugs, has led to resistant forms of all these drugs. Of course, TB is different. I mean, TB is more systematized as far as the treatment is concerned, but other antimicrobials, people have prescribed higher antibiotics for something which can be cured with lower antibiotics, and that's why we have development of resistance. And the worst part is many pathogenic bacteria are able to transfer the resistance to the mycobacteria, because when the granuloma forms, it's not just mycobacteria. There are other bacteria. If you read more about that, you will see that other bacteria also coexist in that particular granuloma. And it is very high possibility that either way the transient genes or other resistant trait happens, so that there is cross resistance. And that's where if you ask any anti-TB researcher, he or she would say that he would not want any drug which will inhibit cell wall biosynthesis, or rather he would not work on any target which is involved in mycobacterial cell wall biosynthesis, because of the obvious problems. So there is no drug which can, nobody can claim in the world that mycobacteria will not develop resistance to this drug. It's just when it happens, because it's like after 40-50 years the new drug has come, and if the resistance develops in 1 year, so you will have to wait till a very long time before we are able to tackle the grave situation, right? So it's like if you are able to delay the development of resistance, so you are actually buying time so that you develop new drugs, new strategies to come up with the anti-tubercular therapeutics. It is bad, actually.
Thank you, sir.
Sir, good morning, sir.
Hi, good morning.
This is K. Raju, sir, working as assistant professor in Sri Vishnu College of Pharmacy, sir.
Yes, please.
First of all, thank you so much, sir, for your valuable information about tuberculosis anti-tubercular drugs, new chemical entities, and new targets, sir.
Thank you.
So I have small doubt about a small doubt, sir. That is, new targets are identified, sir. The drugs, existing drugs like isoniazid and rifampicin can act on new targets, or only new chemical entities.
Well, again, that's a great question. Just like any other drug which can be considered as a part of a drug repurposing campaign. But if you look at, say, particularly isoniazid actually gets converted into a free radical. It's a prodrug. Isoniazid is a prodrug. It gets converted into a free radical, and that free radical can eventually inhibit the INHA, which is response. I think it's involved in cell wall biosynthesis as well. So what happens is mycobacteria gets killed, right? So it is very much possible that isoniazid may act by different mechanism, but it's just that we at this point of time don't know. It's just something which needs to be discovered. I mean, if you read about isoniazid, iproniazid is an antidepressant drug, which was identified as a part of anti-tubercular program. And then eventually they thought that they actually saw that the patients were very calm, and eventually it was repurposed for antidepressant effect, because I think it inhibited MAO A or MAO B. Yeah, so the point is, it's very much possible. It is just that we don't know yet, or there are no testing or no assays which has yet tapped that potential or identified, or from any assay there is some indication that isoniazid might actually be inhibiting other targets. But it is possible. It is very much possible. I mean, it's a reactive molecule, and in fact, if you see, if you look at PubChem bioassay, just take a look at isoniazid monograph, and if you see over a period of time, people have tried to investigate it in many different assays. So it is possible that if not mycobacteria, it might actually work against say some new pathogen. It could be SARS-CoV-2, who knows, right? So it's just a matter of getting it discovered. Yeah.
Another doubt, sir. Is there any side effects for newly identified molecules, sir?
Well, there are like potential side effects. It's like any other drug which essentially can have. I don't know if you know, but then every drug has got on average 70 different side effects, because these are small molecules. They'll go and bind anywhere and everywhere. So we are yet to find out or discover a drug which will be devoid of side effects. So these new entities will also have side effects, but the moment you have got a drug which is acting, or new chemical entity which is acting on a target which is selective or very specific for mycobacteria, then the chances of side effects in some way is less, because it's not inhibiting a similar target in human or in the host. Right. So that's where it's like in case of IMPDH, we know that there is MTB GuaB2, there is human IMPDH which is responsible for cell division or rather similar function. So if my molecule also inhibits human IMPDH, then I have a problem. Right? So that's where one has to be careful. Even if you're working on a target for which there is a counterpart in humans, you have to address the selectivity issues, and that's where your initial like drug lead discovery and lead optimization stages you have to address. You have to make sure that you have address, and in fact we do that for MTB and IMPDH. We always test the molecules which are promising as MTB IMPDH inhibitors or MTB GuaB2 inhibitors, also for human IMPDH inhibition, and so far we have seen they are not. I mean, there were few molecules which are also human IMPDH inhibitors. We did not touch them for further studies.
Yes, sir.
Yeah.
Thank you so much, sir.
Thank you.
Thank you, sir, for your kind answers. I know there are a lot of questions today because the topic was very interesting, and we have tried to solve it out somewhat. Thank you, sir. Now I would like Principal Sir, Dr. Prasad Garu, to speak for today's workshop.
Thank you, sir, Dr. Prasad, sir. You have given very elaborate lecture even the disease is very old. New approaches how we given it, and then how the new targets are approaching towards new targets and the disease. You know, enlightened the link with the TB and other complications which are due to this one. Even though it is old disease, it is a persisting disease in India.
Yes.
The disease still is working well. So I have done very excellent presentation, and then so doubts are also people are asking. Your clarification is more very clear clarification you have given it on behalf of our Sri Vishnu Education Society. I thank you for immediately accepting our invitation and giving your beautiful presentation. So we hope we will be having similar type of programs in future. We will expect same response from your side.
Sure. Thank you so much, sir. Anytime. Thank you so much. I really loved it. Thank you so much.
So I thank all the participants to be here with us. Tomorrow will be the day 3rd. And now I would, Kumar Sai, it is up.
Yeah, continue, please. Continue.
So tomorrow we are meeting. Maybe the time is tomorrow 1 hour previously we are doing it. So I hope all of you tomorrow. Thank you. So
Thank
this
you
is
so
the
much.
end of this is the end of the live stream.
Thank you.
Thanks, Prashant. It's a wonderful session. I really appreciate your patience in taking time and coming